Hello everyone, welcome back to another video from Shomu's Biology. In this lecture, we are going to talk about cholera toxin mode of action. If you want to know the mechanism of action of cholera toxin, then this video is just for you. But before understanding the mechanism of cholera toxin, I want you to remember and understand the exotoxin as a whole. Like what is exotoxin, what are the components of exotoxin, and how exotoxin works and everything. Without knowing that, it will be a little difficult. But still, I'll try to convey my message. If you know what exotoxins are, you know one thing, that the exotoxins are mostly bipartite in nature. They have two subunits to it, right? They have a B subunit and they also have an A subunit. And this B subunit is basically involved in the process of attachment, and so this is the attachment, and A subunit is actually interacting as an active unit that causes the effect or impact on the host cell or tissue. Cholera toxin as a virulent factor released by the vibrio cholerae causes diarrhea, watery diarrhea, right? Whenever the cholera toxin is released our intestine, they targetedly release it to the intestine. The intestinal cells, intestinal epithelial cells are the target of cholera toxins. Because why they are target? Because these toxins work in a very specific target manner, because the B subunit of this toxin will bind specifically to a receptor known as ganglioside receptor, and this ganglioside receptor is particularly found in our intestinal epithelial cells. That's why the cholera toxin always target the intestinal epithelial cells, and it causes the change in osmolarity, and as a result the net flow of water from the intestinal epithelial cells to the intestinal lumen, and that causes diarrhea. And due to this diarrhea, severe water loss, body undergoes dehydration, and ultimately with prolonged dehydration a person can even die. Okay, so let me tell you the simple idea here is that to understand this cholera toxins mechanism, we need to know what goes on in the intestinal epithelial cells. So if I draw as these intestinal epithelial cells like this, these are epithelial cells, and these are the joints between the epithelial cells and all, and this is intestinal lumen. So let me write it. This is intestinal lumen and this is the epithelial cells. Okay, and now I'm also going to draw the effect of the toxin here. So let's imagine that in the cell surface there is a receptor. This is ganglioside receptor, GM1 receptor, and to this GM1 receptor our toxin will bind. So this is B subunit, and in case of cholera toxin this is more than one B subunit out there. More than one B subunits are present. So B subunit here, for simplicity I am only writing as a single B unit, but there are more than one B unit, and then this is A part of the toxin, the active part of the toxin binds to it. And then what happens is that a internalization inside the vesicle. After the internalization is done, let us just go fast in here, and let's just draw this. That after this internalization, simply what we know is that the A and B subunits will be separated. So B subunit is separated, A subunit is also separated. Now we are more concerned about the A unit, because B unit's job is to deliver the cholera toxin inside the cell. That's it. Once the job is done, then rest is history. Means rest job should be done by A subunit, not the B subunit. So what A will do now, once this cholera toxin's active subunit is present in the intestinal epithelial cell, this subunit will cause G protein. Remember, there is G protein present in the cell. In all our cells, mostly you will find G proteins present. The job of G protein is that once G protein gets activated, it can convert ATP into cyclic AMP. Okay, and if more and more cyclic AMP is produced in the cell, this cyclic AMP are going to activate protein kinase A. So this G protein will convert ATP into cyclic AMP. And once the cyclic AMP is produced, this cyclic AMP is going to activate the protein kinase A. Now remember one thing, I am writing this taking two cells, but actually this whole process takes place in individual cells. Not likely that they are contacting between the cells, not likely. So the toxins can come and in contact with individual cells, and the same impact is visible inside the cell of individual cells. But as I'm running out of the page, that's why I'm drawing it with two images. Okay. So once protein kinase is activated, this particular protein kinase, what it will do, it activates a particular ion channel, chloride ion channel, known as CFTR. CFTR, cystic fibrosis transmembrane conductance regulator. Or in very simple words, you can say that this is nothing but a chloride channel. Chloride ion channel. That's it. This channel allows chloride ions to move out. Okay. So once this is activated, chloride ions start to move out. Out where? To the intestinal lumen. Got it? So chlorides start moving out to the intestinal lumen. As the chloride ions start moving out to the intestinal lumen, there is a charge disbalance. Obviously, because chloride is negatively charged, more negative charge ions are moving towards intestinal lumen. So to balance that charge, more sodium ions start to flow via sodium channels. This is sodium channel. Okay. And what will happen? More and more sodium ion start to flow out to the intestinal lumen in order to balance the charge difference. So the first chloride ions start moving, second sodium ions start moving, and now soon so many ions are rushing out of the intestinal epithelial cells to the intestinal lumen. And to balance the osmolarity, to balance osmolarity, the solute concentration will be maximum here in the lumen and minimum to the intestinal epithelial cell cytosol. So to balance this, osmosis. What is osmosis? The movement of water from low solute concentration to high solute concentration. So water will flow, let me draw, water will flow from low to high. From low to high. So water net movement. So more and more water start to be filled where? In the intestinal lumen. And this is the place where everything is happening, where the fecal compaction, the feces and everything is present. So this is more and more water to your intestinal lumen. Bloating. Watery diarrhea. This is how the watery diarrhea will be done, will be caused. Okay? The primary response is done by the cholera toxin, but then activating CFTR, then release of chloride ions, second release of sodium ions, third movement of water for osmotic balance. That causes severe watery diarrhea. And along with that, as the bacteria start colonizing, obviously they are going to damage this interaction between the neighboring epithelial cells. The tight junctions are out there that actually protect us against the invasion of this bacteria. So this is a mechanism of action of cholera toxin causing diarrhea, and they are known as enterotoxin because the target is enteric, the enteric cells of the body. I believe you have a clear idea now. If you like this video, please hit the like button, share this video with your friends, subscribe to this channel to get more videos like that in future. Thank you. Bye.